Okay, we can get started. We were talking about initiation. We just began our discussion of initiation last time. And it's interesting how the same principles come up again and again. Replication, transcription, now translation. There are these distinct phases: initiation, elongation, and termination for all of them. And DNA polymerase can only elongate, it can't initiate. And so a strategy has to be found to provide the DNA polymerase with a complex that already looks like the elongation phase of replication. And that's done, of course, by having an RNA primer, so that the DNA polymerase sees the primer, it looks like an elongation step, and so it proceeds with elongation, and then later on the primer issue is resolved by getting rid of the primer and so forth. We went through that. In RNA polymerase, we have a similar situation where the main activity of the RNA polymerase in terms of what it actually does is elongation, of course. There's initiation, which is fundamentally important in regulation, but from the standpoint of mechanics of RNA synthesis, it's the elongation that the RNA polymerase does most of the time. And we saw how initiation was sort of a tortuous process in which there was this abortive initiation stage where short little oligonucleotides were synthesized and released and synthesized and released until finally it could enter the elongation stage. And we have here a similar issue. The initiation step in translation operates conceptually in a way similar to replication, in that there's a mimicking of the elongation step as the initiation step. So let's look at that. Need to create something. The initiation step of protein synthesis is essentially the creation of something that looks to the ribosome like it's in the middle of elongation. So let's take it here from the top, keeping that fundamental principle in mind. Here is an isolated 30S subunit, which is what we start with when we talk about initiation of translation. Obviously, this 30S subunit was previously part of an intact ribosome with the 30S, 50S ribosome complex with messenger RNAs and tRNAs on it and so forth. But at the initiation step, or in preparation for initiation, after termination has resulted in the dissociation of the two subunits from each other, something that's actually helped by this initiation factor 3, which binds to the isolated 30S subunit to prevent reassociation of another, a premature reassociation of another 50S. So the IF3 is bound here to the 30S subunit to keep the 30S subunit separate from the 50S subunit until these initiation steps, these initial steps of the initiation phase have taken place. So the IF3, the initiation factor 3, helps to prevent premature binding of the 50S subunit at this stage, and at the same time helps a new messenger RNA to bind to the 30S subunit. At the same time, this initiation factor 1 binds in the A site. And that's to prevent premature binding of a tRNA, an amino acyl tRNA, to this A site. Why does the initiation factor 1 want to prevent binding of any amino acyl tRNA to the A site? Because the objective here is to create something that looks like an elongation complex. And so we want to bind something directly to the P site. I reminded you of this before, but remember that the normal sequence of events is that an amino acyl A tRNA binds in the A site. There is attack of the amine nitrogen on the ester bond here of this amino acyl tRNA. With the transfer of this peptide to this amino acid on the incoming tRNA bound initially in the A site, but then it's transferred to the P site, carrying the peptide with it, and another amino acyl tRNA comes in here. So the tRNAs during elongation can only enter at the A site, and they get to the P site only by the messenger RNA with the tRNA associated with it ratcheting one notch over. So that's the only way that a tRNA gets to the P site during elongation. It enters in the A site, and then it ratchets along one notch. Initiation circumvents that by having the initiator tRNA bind directly to the P site in order to mimic this tRNA with a peptide chain. And so IF1 associates here to prevent premature association of any amino-acyl tRNA to the A site. Now, simultaneous with this, we have the preparation of the tRNA that is going to mimic an elongation complex, namely the F-formyl methionine charged F-met tRNA. Here's formyl methionine, and you can see that this looks like a peptide bond here. This obviously is not an amino acid, it's just a formyl group, but this linkage is what is recognized by a lot of the proteins that are involved with guiding the amino-acyl tRNAs around. We'll encounter that. So there's this special F-met tRNA that is charged with a formyl methionine. And it associates with initiation factor 2. And initiation factor 2 helps the F-met tRNA bind directly to the P site. Rather than the A site, which is occupied here. Now, something very interesting happens at this stage. You see that there's, this is the initiator codon. This is the anticodon of the F-met tRNA. But this step and this step can happen in either order. What does that tell you about the role of codon-anticodon recognition for the initiator tRNA? Yeah?
It's not dependent on the binding of the 30 subunit?
Almost. It's the, maybe saying it the other way around. The binding of these things to the 30th subunit is not dependent on this recognition. I think that's what you probably meant, right? So the messenger RNA can bind first, and then the initiator tRNA, which is what one would have naively expected. But remember that this is designed to mimic an elongation complex, and so to get this to associate in the right place on the ribosome, these entities, both the tRNA and the initiation factor, recognize contacts on the ribosome itself and associate in exactly the right spot. So the messenger RNA associates, but it can associate after the tRNA. It doesn't have to be there before, because their juxtaposition is guided by their independent ability to recognize the right place to be on the ribosome. Then after they independently arrive on the ribosome, the codon and the anticodon notice each other, interact, and then elongation is ready to go. The 50S subunit, a conformational change occurs that makes this receptive to the binding of the 50S subunit, which kicks off IF3, which was preventing the 50S subunit from binding previously. So the IF3 comes out. And then these other initiation factors are released, and now the ribosome is ready for normal elongation. The next amino-acyl tRNA can come and bind at the A site, and we're going to talk about how elongation occurs. Yeah?
I was just wondering, what is it the methionine? I'm sorry, the formal group on methionine, is that what allows, the recognition to that site, or is it something else?
It's not this linkage that allows the formal methionine tRNA to recognize the protein. This linkage is recognized by the initiation factor. So you're right, this linkage is recognized in many different contexts. And these proteins are designed to recognize the linkage. In this case, it's the initiation factor that recognizes this linkage. And then the binding to the ribosome occurs by virtue of recognition of both the initiation factor and contacts on the tRNA with the ribosome. This is an important point, the recognition of the formal methionine by the initiation factor and the creation of the formal methionyl tRNA in the first place. There are actually two tRNAs for formal methionine. You know there have to be two tRNAs because we just talked about one tRNA for formal methionine to start translation. But there are AUG codons inside the protein as well, which are recognized by just regular methional tRNA. And the way these two different tRNAs are created is that there are in fact two tRNAs that get charged with methionine. And they get charged with methionine by two different methional tRNA synthetases. So there are two different synthetases that aminoacylate. So the tRNAs get aminoacylated by the synthetases, and then the specificity is generated by a transformylase, which puts the formal group onto the methional tRNA. So the charging step, the attachment of the methionine to either the F-met tRNA or just the methional tRNA, those two different tRNAs, are both charged with methionine, but the formal transferase that puts the formal group onto the methionine recognizes only the tRNA, recognizes only the F-met tRNA, the tRNA that is recognized by the initiation factor, the tRNA that can recognize the P site directly in the ribosome. So that's a very special tRNA. So charging of methionine on these two different tRNAs is not specific. The methionine goes on either the regular methionine tRNA or onto the F-met tRNA, or I should say and onto the F-met tRNA. But the transformylase, formal transferring, the transformylase puts this on only on the methionine that is on the F-met tRNA. So the recognition is of the tRNA, of the particular species of tRNA. Yeah.
You were saying that the 30S subunit can either associate with the mRNA first and then associate with the IF2 F-met tRNA complex, or vice versa. My question is that if the F-met tRNA IF2 complex is not there, or is not on the 30S subunit, and the 30S subunit recognizes the mRNA first, how does it know where to be like on the right position? Because there is no anti-codon-codon interaction, so
How does who know to be in the right position?
the 30S subunit
The 30S subunit to be in the right position of what?
on the mRNA.
On the mRNA. Oh, fabulous, I was waiting for somebody to ask that question. Yeah, you had a, before we go to that, you had a question too?
Yeah, so, you said that there's two different, enzymes that activate the tRNA for the methionine because there's two different, there's
Wait. Did I say that?
That's what I remember.
Oh, no, it's a single, enzyme that charges both tRNAs with methionine.
Okay, and then it's just after when you put on the formal announcements.
That's right, I'm sorry. So if I said that there were two different synthetases, I didn't mean that. I was lying.
Can you repeat that? Can you repeat the whole process, just in there's not two synthetases?
It's a single synthetase that charges both of the methynyl tRNAs. Both the methionine tRNAs and the F-met tRNA, a single synthetase. There's no specificity between the two tRNAs at that stage. The specificity arises, thanks for catching that. The specificity arises by virtue of the transformulase, which recognizes only the F-met charged tRNA and formulates that. And then that is recognized by the GTP, by the IF2. Okay, so, yeah, there's this problem, which actually has two parts, and I mentioned only one part. The messenger RNA can associate with the 30th subunit, and the F-met formal methynyl F-met tRNA, IF2 complex, can associate with the 30th subunit independently. And we talked about how this recognizes the 30th subunit, but we haven't talked about how the messenger RNA knows where to sit down so that these two are in the right register. So here we have the 30th subunit. The messenger RNA is in the right place on the subunit. The tRNAs are not shown, but we know that the formal methynyl tRNA is going to be here at the P site. And we know that the initiation codon of the RNA has to be here. How does the messenger RNA know to be bound to the ribosome in exactly this register? Well, over here, on the RNA, there is something called the Shine-Dalgarno sequence, named after the people who discovered it. You see the sequence here. And the 3' end of the 16S RNA, which is the RNA that comprises the 30S subunit, has a little segment that is complementary to this Shine-Dalgarno sequence. And all messenger RNAs have, in E. coli we're talking about, have this Shine-Dalgarno sequence in them. And that guides the messenger RNA to bind to the 30S subunit with its Shine-Dalgarno sequence in complex with this spot on the 16S RNA, which puts it in exactly the right register. Because the distance between the Shine-Dalgarno sequence and the AUG is almost exactly as shown here, so that it binds the Shine-Dalgarno sequence complement in the 16S RNA, and that places the initiation codon directly over the P site. Now, when you think about regulation, this would be a wonderful exam question, actually. Think about transcription and the regulation of transcription. And we talked about the different elements that are recognized by a polymerase. The -10 region in E. coli, the -35 region, the up region, the et cetera, et cetera. And we talked about how an efficiently transcribed promoter will have very precisely matched sequences and spaces between the sequences, and then the promoter will be very efficient. Otherwise, you can have a promoter that does not have good minus 10 sequences or minus 35 sequences, and it will be an inefficient promoter, which is then subject to regulation, which can be regulated. It can be upregulated by various regulatory programs. Exactly the same thing happens here. Suppose you have a messenger RNA that has a poor match to the Shine-Delgarno sequence. That messenger RNA is going to attach itself to the ribosomes very inefficiently. Or suppose it has a great Shine-Delgarno sequence, but the spacing isn't right. It's going to interact inefficiently with the tRNA anticodon for the tRNA that's located here. And so this, just as in transcription, gives rise to all kinds of possibilities for regulation. Factors that enhance the tightness of binding can be used to upregulate translation for RNAs that have a poor, either a poor match to the Shine-Delgarno or an inappropriate spacing here. So these same principles come up over and over and over again in all different contexts. I just want to make a brief aside here to point out that initiation follows strikingly different principles in some respects in eukaryotes. Eukaryotes do messenger RNAs, eukaryotic messenger RNAs do not have a Shine-Delgarno sequence. But as we've seen, the RNAs in eukaryotes have caps on their 5' ends. E. coli messenger RNAs do not have caps on their 5' ends. The eukaryotic messenger RNAs bind to the, well, it's 40S in eukaryotes because everything's bigger. The 5' cap of the eukaryotic messenger RNAs bind to the 40S ribosome via these various translational factors. And the ribosome actually scans along the messenger RNA looking for the first AUG. And so almost everything that we've talked about occurs differently in eukaryotes. There isn't a precise orchestration of assembly guided by sequence, as we talked about for E. coli, but instead everything initiates with the cap. The ribosome scans along here and it recognizes the AUG. So in this case, the ribosome is actually directly recognizing the initiation codon. And then translation begins. And that's an interesting aspect that can be regulated in eukaryotic cells as well, because the ribosome begins translation at the first codon. But if binding affinity around the first codon is weak, it might skip the first codon and go farther and pick up the second AUG, and so forth. So all of these things are subject to regulation. This whole complex is stabilized here, you can see, by looping around and binding the ribosome at the other end as well via the 3' poly A tail. So the processing of the RNA, the putting of the cap on one end and the putting of the poly A tail on the other, are both involved in translation in eukaryotic cells. So I just wanted to point out these striking differences in strategy between eukaryotes and prokaryotes. But from now on, we're going to continue talking about the prokaryotic situation, and we won't be talking about the eukaryotic situation in any depth. Okay, so now we're all set up for elongation. This is the elongation cycle. There's a lot going on here. So what I'm going to do is I'm going to go through this cycle several times. We'll start by just going through it quickly to see what the major elements are. Then we'll go through and examine some of the steps in more detail. And then we'll go through it a third time, looking at additional steps in even greater detail so that we can sort of build our understanding of the elongation of translation in layers, so to speak. So if you just look at the overall sequence of events here, you can see that after the initiation step, there is a formal methionine tRNA bound here. There's nothing here because, of course, we're initiating, so there's nothing left over from a previous cycle here. So this is empty. And there's something here that's mimicking and elongating polypeptide, and elongation involves the arrival of a new amino acyl tRNA into the A site, guided by an elongation factor, EFTU in complex with GTP. We'll talk about that. This binds in two stages. That's very important. We'll talk about that too. First, in the 30th part of the A site, and subsequently in both parts of the A site. So you notice these boxes. These boxes refer to the, of course, A site, P site, and the exit site. But as we've seen before, there are components of the A, P, and E sites that reside on both the 30S and the 50S subunit. So when we go through our discussion here, we're going to be talking about the different components, both the 50S and the 30S component of the A site and the P site and the E site. So that comes up very soon. You can see that we're going to get complicated over here. So the tRNA associates with the 30S component of the A site initially. This large elongation factor complex prevents it from fitting into the top part, into the 50S part of the site, but after the dissociation of this elongation factor, now the amino acyl tRNA fits nicely into both components of the A site, and peptidal transfer occurs from this tRNA, which carries the growing peptide chain, to this one. So it's transferred over here, and then there's this two-step ratcheting of the ribosome along the messenger RNA, because now we have the polypeptide, or the growing polypeptide, on the newly arrived tRNA, but it's in the A site. And so the messenger RNA has to move along carrying this tRNA with the polypeptide attached to the P site where it belongs. It has to be in the P site in order for the arrival of the next amino acyl RNA to bring in a new amino acid. And so it does ratchet along in two steps again. First the top part, and then the bottom part to arrive back at something like that. And remember here we have the messenger RNA, it's bound to the 30S subunit, and so the bottom parts of the A, P, and E site are the parts that have the messenger RNA. The 3' end of the messenger RNA is over here on the right, and the 5' end is over here. And so what we're talking about when this goes to this is that the messenger RNA has moved one notch over, carrying this codon associated end of the tRNA with it from the A site to the P site. So we're gonna go through this again now, but we're gonna look at it in a bit more detail. Looking at the various steps, why they occur, how they occur. Okay, so here we are at step number 1. This elongation factor, TU, in complex with GTP, is associated with the aminoacyl tRNA. We'll talk about how this association occurs in just a moment. But it is in association with the aminoacyl tRNA, and it looks like this. Can you tell which part of this is the tRNA and which part of it is the protein? Is the tRNA part gray or is it green? Green, yes, of course, because you see the nice helix here, you see the shape of the tRNA, and here is the EFTU bound to this part of the tRNA. This is an elongation factor that's completely general. It has to work with all tRNAs, and so it binds to residues in the tRNAs that are constant from one across all the tRNAs. So it's recognizing the same spots on the tRNA, and it recognizes the aminoacyl linkage, as we talked about. That linkage has to be recognized so that it does not associate with free tRNAs that are not charged with an amino acid. And then there has to be a room in the binding site for the amino acid itself, which is here attached to the 3' end of the tRNA up there. And so it actually has a pocket, a big old pocket that can accommodate any amino acid. So there's space for any amino acid here. And this then is what sticks into the 30th part of the A site. And this is where codon, anticodon recognition takes place. We'll talk about that in the way the codon anticodon recognition takes place next time. But you can see today that occurs first, and then after hydrolysis of the GTP to form EFTUGDP, this is released EFTUGDP, hydrolysis of this GTP to form EFTUGDP, still associated with the ribosome. Notice how the phosphate comes off first, and this comes off later, so there's space here. There's time during which the elongation factor remains bound to the tRNA before being released. This is very important. We're going to talk about it in more detail next time, but you can see that what's happening is that the binding of the aminoacyl tRNA to the A site takes place in 2 steps, thereby providing 2 opportunities for recognition to take place. I don't know if you've ever experienced this, but when I'm writing a manuscript or something, or an email, whatever it is, and if I need to be very sure that I have not made that there are no typos, that I haven't missed any words, I don't have any double words, whatever, and so I want to be very sure that this very important manuscript or email is completely accurate, I'll proofread it. But you must have experienced proofreading things like this, and your brain just skips over missed ands, thes, and so forth, or double ands and double thes, and certain misspellings. Your brain just skips over it. You can read it through several times, and your brain will skip over it every single time. I can't explain why that happens, but it obviously depends on context. For whatever reason, in that particular context, your brain just fills in the gaps. So the proofreading is not 100% accurate. The way I get around that, there are various ways. Sometimes I just change the formatting on the screen so that the lines break at different positions. Or sometimes I print it out and I read the hard copy instead of reading it off the computer screen. Whatever I do, as soon as I change the context, then these errors that I previously was completely oblivious to, they jump out at me. So the principle here is from a conceptual point of view, very, very similar. If you proofread the same thing a second time, the errors that your brain skipped the first time, it's going to skip the second time. So you don't accomplish anything by just proofreading over and over again. Instead, you change the context and then proofread again, and now you pick up, you have a good chance of picking up all of the remaining errors. And that's exactly the strategy that's used here. The EFTU GTP binds. The first time there's codon anti-codon recognition here, but it's strained. It's not the way it should be. Still, because of that strain, the recognition has to be pretty good in order for the match to even take place in the first place. The association here cannot occur because of the association of EFTU GTP with the top part of the RNA. So it can't get into the top part, to the 50S part of the A site. But after hydrolysis of the phosphate, the EFTU changes conformation dramatically. Now the tRNA, because of that dramatic change in the conformation of the EFTU, changes position. And now there's a new possibility for the ribosome to assess the correctness of binding. Because if the binding is incorrect, it's going to interact with the new position in a different way and give the ribosome a new opportunity to proofread. And so that overcomes the thermodynamic limitation that I talked about before, about, for example, with the threonine, alanine 100-fold accuracy limitation. There, the proofreading step was an entirely separate step, which involved hydrolysis off of the valine. Did I say, I think I said alanine. Valine threonine is what I was referring to. The valine is hydrolyzed off in the editing step. But here, in the growing polypeptide chain, you don't want to lose the growing polypeptide by hydrolysis. And so instead, if an error is made, so instead, this very same entity is subject to proofreading in two different steps. And we're going to talk about in more detail how those recognitions occur next time. But there are two steps. And so this is very accurate. After the two proofreading steps, then the EFTU GDP is released. The tRNA is here, and the GDP has to be replaced by GTP. Now there's a problem, because upon hydrolysis of phosphate, the GDP causes the conformational change to occur in the EFTU and in its association with the ribosome because it binds very tightly. It binds very tightly in a different way, and that's what drives the conformational change in the protein. The tightness of binding of the GDP to the EFTU in a way that GTP could not bind. So, if EFTU binds GDP very tightly, more tightly than the GTP, how are you going to get GTP in there? Are you going to affect this exchange? And so this whole cycle is designed to accomplish that. Here the elongation factor TS associates with EFTU GDP. And elongation factor TS associates with the EFTU and has a little projection, a little finger that gets into the GDP binding site. Now we've talked about how nucleotides need to associate with magnesium at virtually all times in order to provide some charge neutralization to the phosphates. And so the binding site of EFTU for GDP, like all such binding sites, has a binding site for the magnesium, as well as for the GDP itself. And that little finger of EFTS wiggles into where the magnesium is binding, altering the balance of interactions, destabilizing the binding of magnesium, and thereby destabilizing the binding of the entire GDP. So now the GDP is bound much less tightly to the complex because of the presence, the destabilizing presence of EFTS, and the GDP comes off and the EFTS now remains. Eventually the EFTS dissociates, and now GTP can come in. But why does GTP come in rather than GDP? What happens if GDP were to bind again? You just have the same problem, you go around in circles, right? So GDP binds tighter, you have to overcome that using EFTS. But then to make sure that GTP gets in, GTP is at 10 times the concentration of GDP in the cell. And so after the EFTS dissociates, what's more likely to get in, GTP or GDP? GTP. Yeah, GTP, because its concentration is so much higher. But the GTP binds less tightly than the GDP, right? That's still true. So this is a very interesting example of the trade-off between stability and kinetics. Here there was a stability issue that had to be overcome. Here it's kinetics that ensures that the GTP binds. So you say, well, sure, but we know that GDP binds more tightly, so obviously it's going to replace the GTP. Well, not so fast, because this is fast. The GTP binds, and before it has time to dissociate, an amino acyl tRNA binds, forming the EFTU GTP amino acyl tRNA complex, which changes the binding of the amino acyl tRNA, changes the conformation of EFTU, so that now it binds GTP tightly. So this is kinetically driven here. The GTP comes in much faster than the GDP can, even though it doesn't bind as tightly as the GDP. But before it can dissociate, an amino acyl tRNA comes in and changes the conformation of the complex so that it now binds GTP tightly and the whole thing can go again. All right, so now we have transpeptidation that must take place. Now, obviously, transpeptidation, this attack of this amino group on that carbonyl carbon is not going to take place when they're 100 miles apart. Instead, this is really what's going on. The two 3' ends of the RNA are juxtaposed very closely in that single catalytic center that I've been talking about previously. And peptidal transfer to this tRNA takes place. So this tRNA is bound in the A site, but it's twisted around so that the two 3' ends are right next to each other. Okay, so now we have this situation, and we have to move this peptidal tRNA over to the next swap. And It turns out that the 30S subunit of the ribosome is constantly rotating, about 6 degrees. So it's constantly oscillating like this. And when it oscillates, obviously the 50S part and the 30S parts of the binding sites are going to be moving like this as this oscillates. So it's going like this. And this tRNA sitting here, when this oscillates and the 30S subunit goes over, look at this first. If you're looking at this, and the 30S part of these binding sites moves over because of this oscillation, so it moves over like that. This is designed to be an unstable relationship with this moved over and this still bound here. And so when this moves over, this flips into the P site. And then we'll discuss next time how EFG rectifies the situation here so that the top end, so that the bottom end